Hello everyone, once again welcome to this series of lecture on biochemistry. So today I'll be talking about metabolism of other hexoses. So once again, I'm Dexter Pajarito. In our previous lecture on glycolysis, we have seen that glucose is the main molecule that enters the metabolic pathway. In this lecture, we will see how galactose, fructose, and mannose enter the glycolytic pathway. So let's begin with galactose. Galactose is brought into glycolysis by first converting into galactose 1-phosphate. The source of phosphate here is ATP. As you can see, ATP gets converted to ADP. And the enzyme that catalyzes this reaction is galactose kinase. The galactose 1-phosphate is converted into UDP galactose. So here, there is a participation of UDP galactose. The uridine diphosphate of glucose now goes to galactose, forming UDP galactose. This reaction is catalyzed by an enzyme, galactose 1-phosphate uridyl transferase. The UDP galactose isomerizes to UDP glucose, and this process is catalyzed by UDP galactose 4 epimerase. The UDP glucose is acted upon by an enzyme, UDP glucose pyrophosphorylase. This enzyme cleaves the pyrophosphate from UTP, and the phosphate group is brought to glucose, forming glucose 1-phosphate. This UDP glucose is a precursor for the synthesis of glycogen via glycogenesis. The glycogen further can be catabolized or broken down by phosphorylasis to form glucose 1-phosphate. The glucose 1-phosphate can be isomerized to form glucose 6-phosphate by the action of phosphoglucomutase. This glucose 6-phosphate can be converted into glucose by the action of glucose 6-phosphatase. So the glucose 6-phosphatase is an enzyme that actually removes the phosphate from the glucose on carbon number 6, generating glucose. Moreover, the glucose 6-phosphate can also be isomerized into fructose 6-phosphate by an enzyme glucose 6-phosphate isomerase. This fructose 6-phosphate actually comes from the phosphorylation of fructose. At this point, we can see the entry of fructose in the glycolysis. So, the fructose is acted upon by hexokinase. This is present primarily in muscle and in adipose. So, phosphorylation of fructose requires ATP to form fructose 6-phosphate. So, at this point, we can see the participation or the entry of mannose in the glycolytic pathway. Mannose is one of the hexoses. So, just like the rest, mannose is phosphorylated by the action of hexokinase. So here, the source of phosphate is also ATP. One of the phosphates of ATP goes to carbon number 6 of mannose, forming mannose 6-phosphate. Then, the mannose 6-phosphate isomerizes to form fructose 6-phosphate by the action of phosphomannose isomerase. At this point, we can see that the fructose 6-phosphate that we have produced earlier can be converted into fructose 1,6-bisphosphate by the action of phosphofructokinase 1. Moreover, this fructose 1,6-bisphosphate can also be acted upon by phosphatase to form fructose 6-phosphate. Now, we can see here that the fructose 1,6-bisphosphate is the intermediate that enters the glycolysis. By the action of aldolase, the molecule of fructose 1,6-bisphosphate is cleaved into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. We know that the dihydroxyacetone phosphate isomerizes to form glyceraldehyde 3-phosphate by the action of triose phosphate isomerase. This TPI is the triose phosphate isomerase, and we know that this enzyme catalyzes reversible reaction. This glyceraldehyde 3-phosphate will eventually become pyruvate along the process of glycolysis. Now, at this point, here we have fructose that is acted upon by fructokinase. This enzyme differs from the enzyme, the hexokinase that we have seen earlier. This fructokinase is present in the liver and it only acts when fructose is in abundance. So, the fructose is phosphorylated by fructokinase and it utilizes ATP in the process. The product is fructose 1-phosphate. This fructose 1-phosphate is cleaved by an aldolase to form glyceraldehyde and dihydroxyacetone phosphate. Notice that glyceraldehyde is not phosphorylated. So therefore, in order for the glyceraldehyde to get into glycolysis, the glyceraldehyde is phosphorylated again by glyceraldehyde kinase, and the source of phosphate is ATP. So the phosphorylated glyceraldehyde becomes glyceraldehyde 3-phosphate, which again eventually goes into glycolysis, then it will become pyruvate. Alright, thanks for watching this presentation, and if you like this video, please consider subscribing to my channel. See you on the next video.